السلام عليكم. إن شاء الله راح نناقش Biochemistry of neurotransmitters بمحاضرة هاي. So طبعا the main resource is the lecture itself. You can go back to Mark's Basic Medical Biochemistry, the 6th edition. في 10 pages that nicely describe the process. لكن I advise you to go to this website as well. في good information in terms of anatomy, physiology, and biochemistry that you can refer to as well. So first we need to start defining and talking about the characteristics of neurotransmitters. You will notice that there are exceptions, but these are the main characteristics and features of neurotransmitters. So basically, a neurotransmitter is considered as such if it is synthesized and stored in a presynaptic neuron. The enzymes that are needed to synthesize these neurotransmitters exist in the presynaptic neuron as well. The neurotransmitter بعدين بيصير له release into outside out of the nerve cell, the presynaptic neuron, to the synapse following depolarization of the nerve, and it is usually associated with influx of calcium ions. So the calcium ion concentration goes up really high. Once it gets released, it can bind the postsynaptic cell to a receptor على the surface of postsynaptic cell. أو ممكن it can bind to receptors على presynaptic terminal as well. As a result of this interaction ما بين ligand, the neurotransmitter, مع receptor, بيصير في عنا signal transduction taking place inside the cell. The remaining neurotransmitter is inactivated. It's removed in order to go back to the relaxing stage. So there are different types of neurotransmitters depending on their structure. فعنا small molecule neurotransmitters وبرضه في عنا sub classification لهدول النيوروترانسميترز. فعنا biogenic amines زي الابنفرين, dopamine, histamine, serotonin, and so on. And these are synthesized from amino acids. في عنا as well amino acids that can act as neurotransmitters, as well as a modified amino acid اللي هو الجابا. Acetylcholine, and we'll talk about these three classes. بعدين في عنا ATP can also bind to a receptor inducing a signal. We're not going to talk about ATP. The second class is neuropeptides, and then we have gases like carbon monoxide and nitric oxide. We'll talk about nitric oxide only. كانوا in the past يعتقدوا بأنه every neuron has its own small molecule neurotransmitter. طيب كانوا يعتقدوا إنه في كل نيورون في له or group of neurons في لهم small molecule neurotransmitter only and a neuropeptide only. لكن it turns out recently إنه لا والله في عنا إحنا neurons that can contain more than one small molecule neurotransmitter. We have neurons that have a combination of small molecule neurotransmitters with neuropeptides or more than one neuropeptide as well. هلا كيف يصير في release ل one versus the other؟ هون research that is going on, and it has to do with the level and probably localization of calcium ion influx into the cell. So this shows the different structures of these neurotransmitters, the different classes. So we have these amino acids. We have monoamines like catecholamines, serotonin, histamine as well. These are the neuropeptides, so they are composed of amino acids, okay, and they are small peptides, and ATP as well. So there are different classes and they have these structures. So we'll talk about these molecules right now. Now, there's something important in terms of differences between neuropeptides and small molecule neurotransmitters, and you do need to know these differences. Okay, and we'll talk about some of them at least throughout the lecture. So in terms of neuropeptides, they are short chain peptides. They're composed of 3 to 60 amino acids. They can have large molecular weight. Small molecule neurotransmitters, well, you can tell from the name, they are small. The neuropeptides are slow acting versus these are fast acting. Neuropeptides have a slow response versus acute response for small molecule neurotransmitters. Neuropeptides have prolonged action, but small molecule neurotransmitters have short-term response. Neuropeptides can act on several receptors, so you can have one peptide binding to different receptors, but for small molecule neurotransmitters, they have their own specific receptors. Neuropeptides can change metabolism, but not necessarily for small molecule neurotransmitters. Neuropeptides can change gene expression. Here, most of them do not, but we've noticed that some of them do as well. Okay, but most do not. Neuropeptides are synthesized in the endoplasmic reticulum and the Golgi apparatus, but small molecule neurotransmitters, they're mainly synthesized in the presynaptic nerve terminal itself, mainly in the cytosol. There are some exceptions ورح نحكي عنهم إنه they are synthesized inside vesicles. Neuropeptides are synthesized in low concentrations, but for small molecule neurotransmitters, they are synthesized in high concentrations. Neuropeptides are found all over the neuron, but small molecule neurotransmitters are found at the axon terminals of presynaptic neurons. Neuropeptides are stored in large dense core vesicles, and I'll show you an image of these vesicles. Small molecule neurotransmitters are stored in secretory vesicles, and again I'll show you an image of those vesicles. Neuropeptides are released at low increase of cytosolic calcium ion concentrations, but small molecule neurotransmitters require higher cytosolic increase, higher increase in cytosolic calcium ion concentration. Neuropeptides have different sites of action than their origin, but small molecule neurotransmitters usually they act in the vicinity, and in the neighborhood of where they are released. Neuropeptides are not taken up by the presynaptic neuron, and they are not reused, but small molecule neurotransmitters can be recycled. They can be taken up again by the secreting presynaptic neuron, and they can be repackaged inside vesicles and released. Neuropeptides are more potent than small molecule neurotransmitters, and they are inactivated by proteolytic degradation or diffusion outside or away from the secreting cell. Small molecule neurotransmitters are terminated by uptake by neighboring cells like glial cells, by diffusion, or by enzymatic degradation. So I'll briefly talk about the neuropeptides, and this is for your own information. Okay, يعني أنا ما بدي إياكم طبعا تروحوا عليهم تحفظوهم and so on. No, بس هذه يعني reference لكم. You can visit هدول الويب sites to learn more about neuropeptides. في عندنا مجموعة حاليا كبيرة من النيوروبيبتايدز والفانكشنز تبعهم كتير variable. اوكي، يعني في منهم لهم دخل بالبيهيفير، في لهم بالبين perception، في منهم بيتحكموا او كنترول الميموري او الابيتايت، العطش، التمبرتشر، الفيلينجز in general, the sleep and so on. يعني variable. اوكي. some of them ممكن يلعبوا دور كهرمونز أو كنيوروترانسميترز، ف يعني عندهم dual function. they're synthesized just like any other protein، يعني بصير لهم transcription، بعدين translation على mainly بالendoplasmic reticulum، بصير لهم processing. المسنجر RNA ممكن يصير له post transcriptional regulation. طبعا هنا transcriptional regulation the level of transcription can be controlled. لما يطلع المسنجر RNA ممكن يصير له alternative splicing. البروتين expression ممكن يصير له control as well و regulation. بعدين عندنا protein processing post translational modification للبروتينز، من ضمنهم الcleavage والglycosylation and so on. They can be tissue specific and there are a number of examples مثل substance P, neurokinin A and so on. طيب هلا I'll just give you an example for a couple of neuropeptides وكيف بيصير لهم modification. نبلش أول شيء بهذه بالpost transcriptional processing of neuropeptides. عندنا substance P ونيوروكاينين A. هدول الاثنين بيجوا from the same gene. They are synthesized from the same gene. لكن شو بيصير عندنا هون؟ بيصير عندنا alternative splicing. فبالnervous system في عندنا messenger RNA mature messenger RNA بطلع لنا substance P. لكن بالnervous system والperipheral tissues بصير alternative splicing للmessenger RNA بحيث انه يطلع لنا neurokinin. So this is just to give you one example. Another اللي هو pro-opiomelanocortin peptide, polypeptide. It's a large peptide اللي بيصير لها synthesis بالpituitary gland أو بالpituitary gland in different أو الhypothalamus. طيب بالpituitary gland بالanterior lobe of the pituitary gland اللي بيصير عندنا هون إنه بعد ما يصير في translation or synthesis of the polypeptide بيصير في عندنا cleavage of the polypeptide into different hormones, different peptides that can act as hormones. هلا بالhypothalamus بالskin pars intermedia of the pituitary gland برضه بصير عندنا synthesis of the same polypeptide وبرضه بصير عندنا similar processing لكن بصير في further processing, further cleavage of these peptides into even shorter peptides and they have different functions. I do not want you to memorize these different peptides. Okay, بس بدي إياكم تعرفوا كيف ممكن يصير في synthesis or processing للنيوروبيبتايدز. هلا فعلشان هيك احنا عمالنا نتكلم على regulation at different levels. ممكن يصير at the by transcriptional factors. يعني ممكن يصير at the transcriptional level by transcriptional factors أو الهيستون modification اللي هو epigenetics اللي هو methylation of DNA أو modification chemical modification of histones الchromatin structure اللي هو الeuchromatin والheterochromatin لما يكون packed أو لما يكون loose. ممكن يصير في post transcriptional regulation processing processing للمسنجر RNA يعني splicing. ممكن يكون control على processing modification of the RNA the transport of the RNA. ممكن يصير في binding لمايكرو ار ان اي على المسنجر ار ان اي ويصير في الاستابيليتي تبعت المسنجر ار ان اي would be affected, as well as the localization of the messenger RNA. وين راح يروح هذا المسنجر RNA بالcytosol. This is another level of regulation that is even يعني higher or more sophisticated, and so on. A translational regulation as well, وبعدين عندنا post translational regulation, modification of the neuropeptides زي again glycosylation, cleavage, and so on. فعندنا anyhow regulation at different levels. طبعا لما يصير في packaging للنيوروبيبتايدز ب large dense core vesicles, برضو regulation للسكريشن، وبس يصير في secretion حيصير عندنا regulation outside the cell, extracellular regulation, diffusion, proteolytic cleavage, degradation of the neuropeptides, and so on. فاحنا حكينا قبل هيك انه actually the effect of duration of action of neuropeptides can be prolonged compared to small molecule neurotransmitters. طيب بس كيف بيصير في عنا اذا synthesis للنeuropeptides. So basically synthesis طبعا starts in the nucleus, transcription, بعدين translation على surface of the endoplasmic reticulum. فبكون للنيوروبيبتايدز هي عبارة عن pro peptide. البري region البري sequence هي بأول 70 amino acids من البيبتايد اللي هي directs the peptide على ال surface of the rough endoplasmic reticulum اللي بكون عليه ribosomes. As the peptide goes into the ER, as it is translocated into the ER, the pre-region is cleaved. فبضل عندنا the pro-peptide. Pro-peptide يعني inactive peptide. لسه the protein is not active, whether it is an enzyme or a hormone. Pro-peptide معناته inactive peptide. But من ال ER it goes into the Golgi apparatus, بيصير في عنا modification of the peptide, and then they are packaged into large dense core vesicles اللي هم هدول. This is a large dense core vesicle, هذه large dense core vesicle, and so on. And they are transported with the modifying enzymes via fast axonal transport. Okay, fast axonal transport لأنه بالsmall molecule neurotransmitters حين عندنا process تاني اللي هو slow transport of these vesicles. طيب during the transport of the neuropeptides بيصير في عندنا further modification of these peptides, cleavage for example, فبكون عندنا the peptide hormones inside these vesicles, and then they are released gradually with the influx of calcium ions into via channels into the synapse. The action is prolonged and they bind to G protein coupled receptors, and that's how they mainly function. وزي ما حكينا انه once they are in the synapse, some of them can go into the bloodstream and they're transported far away from the site of secretion. بصير في عنا diffusion and degradation, proteolytic degradation of these neuropeptides. طيب. Calcium ions are needed for the fusion of these vesicles with the plasma membrane. لكن في عنا two important points بالنسبة للكالسيوم influx. أول شيء إنه the site of calcium entry, calcium ion entry into the cells it can be far away from where these vesicles are localized. طيب. And the concentration that is needed to let the vesicles fuse with the plasma membrane وبعدين يصير لنا release is lower compared to the concentration of calcium ions needed for the release of small molecule neurotransmitters. So keep that in mind. طب خلينا نتكلم عن السمول مولكيول نيوروترانسميترز هلا. فنتكلم عن synthesis of these small molecule neurotransmitters. هلا ديروا بالكم من هاي النقطة انه السينثيسيس هي بتصير للانزيمز that are needed for the synthesis of small molecule neurotransmitters زيها زي بقية البروتينز. بصير في transcription، بصير في translation، modification بالاندوبلازميك ريتيكولم بالجولجي، وبعدين هدول الانزيمز they are transported along microtubules via slow axonal transport لوين؟ للcell periphery، اوكي، للبريسينابتيك terminal. What happens then انه هذه بصير في عنا synthesis للنيوروترانسميترز in the cytosol and the by these enzymes that transported enzymes، وبعدين بصير في packaging للنيوروترانسميترز inside secretory vesicles, small secretory vesicles, and they wait for the influx of calcium. Calcium goes in لما يكون عندنا stimulation، لما يكون عندنا depolarization بيصير في عندنا entry of calcium ions into the cells and the vesicles fuse with the plasma membrane releasing the neurotransmitters. هلا بالسينابس what happens then انه لازم يصير في inactivation. النيوروترانسميتر would bind to a receptor موجود على presynaptic nerve surface, stimulation of a signal and so on. بعدين لازم يصير في inactivation, and this inactivation takes place by different mechanisms. منها reuptake أو diffusion, okay, diffusion in the synapse, synaptic cleft. بيصير في reuptake, so it goes back into the presynaptic neuron and it can be packaged again into vesicles and released. ممكن يصير في uptake by the neighboring cells, like glial cells, and the postsynaptic nerve. وممكن يصير في inactivation, enzymatic inactivation of these neurotransmitters. هلا in terms of calcium ions, the influx, the site of influx of calcium ions بكون nearby where these secretory synaptic vesicles are located inside the presynaptic terminal, presynaptic nerve. The influx is really high. That is, the concentration really increases, and it goes up from 0.1 micromolar to 50 to 100 micromolar. This is huge, sudden, acute increase in the concentration of calcium ions. So compare this to neuropeptides. This is really nice image of these vesicles. Again, this is the large dense core vesicle that encompasses or that contains the neuropeptides. هدول هم السكريتوري vesicles الصغار موجودين كلهم accumulated near the cell surface, waiting for the influx of calcium ions. And this just it shows you how they are lining up. Actually في عندنا هون space بين الفيسيكل مع البلازما membrane، فعشان هيك ما عندنا fusion بين الفيسيكل والبلازما membrane. بس أول ما الكالسيوم يدخل بيصير في عندنا modification وفي bridging بين الفيسيكل مع البلازما membrane، أو يعني the involvement of many proteins اللي بيخلوا الفيسيكل attached للبلازما membrane. بعدين بيصير في عندنا fusion of the vesicle مع البلازما membrane, release of the neurotransmitters is followed, and so on. Okay, that basically what happens, and this is what calcium ions do. خلينا ندرس examples of small molecule neurotransmitters. راح نقضي وقت كويس على هدول الثلاث لأنه they are well studied. We have a wealth of information regarding هذول small molecule neurotransmitters. Well, regulation applies to other neurotransmitters as well, small molecule neurotransmitters. So, هدول the synthesis of these neurotransmitters اللي هم dopamine, norepinephrine, and epinephrine, their synthesis requires a number of cofactors اللي هم S-adenosyl methionine. فعندنا methyl transfer. طيب، وهذا الميثيل transfer requires folate وفيتامين B12, so keep that in mind. Pyridoxal phosphate is needed as well, or vitamin B6 for transamination with decarboxylation. Tetrahydrobiopterin is another cofactor, another organic cofactor. Tetrahydrobiopterin يمكن ما سمعتوا فيه قبل هيك، لكن it is synthesized from GTP and it exists as dihydrobiopterin or tetrahydrobiopterin. Do not memorize this synthetic pathway, by the way. This is the structure of the tetrahydrobiopterin. Synthesis of these three neurotransmitters takes place in the liver, or we can get it from diet, and we can synthesize it from phenylalanine by phenylalanine hydroxylase. فبصير في hydroxylation of this tyrosine, and then tyrosine can enter the neurons by active transport. Now, phenylalanine is an essential amino acid. We have to get it from diet, but once we do, we can synthesize tyrosine. عشان هيك tyrosine is known as a conditional essential amino acid, أو conditional non-essential amino acid, لأنه we get it from an essential amino acid, but once we get the essential amino acid, that is phenylalanine, we can synthesize tyrosine. So that's reaction number one. Then tyrosine is hydroxylated again by tyrosine hydroxylase into an intermediate known as DOPA, and this is a rate-limiting reaction because tyrosine hydroxylase is highly regulated, so it's a slow reaction, and it requires tetrahydrobiopterin. Tetrahydrobiopterin is needed for hydroxylating rings. Okay, so عشان هيك احنا بدنا tetrahydrobiopterin. ما علينا. فعندنا هذا الانترميديت اللي هو دوبا. هذه الرينج اسمها كاتيكول. عشان هيك هدول الثلاث نيورو ترانسميترز يعرفوا باسم كاتيكول امينز. كاتيكول امينز. ما علينا. فعندنا هذه الدوبا. هذا الرياكشن بيصير بالسيتوسول. برضه بالسيتوسول بيصير عندنا الرياكشن اللي هو decarboxylation of دوبا عشان نعمل دوبامين. وهذا الرياكشن decarboxylation it requires vitamin B6 أو pyridoxal phosphate. What happens then إنه الدوبامين هذا بيصير له packaging inside vesicles. okay, وبالvesicles بتحول إلى norepinephrine. والnorepinephrine بعدين بيطلع لبرا الvesicles and it gets converted by methyltransferase enzyme into epinephrine اللي هو عندنا الميثيل جروب هاي اللي موجودة عندنا هون. طيب، فعندنا hydroxylation فحطينا hydroxyl جروب هون، بعدين عندنا decarboxylation شلنا هاي الجروب، بعدين عملنا another hydroxylation reaction اوكي حطينا الهيدروكسيل جروب right here، وبعدين عملنا methyl transferase methyl transfer reaction حطينا الميثيل جروب right here. هذا هو الابينفرين. طيب ناخذهم واحد واحد هلا، ناخذهم واحد واحد. So tyrosine بيصير له conversion لدوبامين via دوبا. Dopamine is then packaged بصير الرياكشنز هذه بالcell periphery. بصير packaging للدوبامين inside vesicles, and these vesicles wait for the influx of calcium ions and they fuse with the plasma membrane, releasing dopamine outside. Dopamine binds to a receptor على postsynaptic nerve, and then it has to be inactivated, and it gets inactivated by one, reuptake. Most of it would be taken up again by the presynaptic neuron, and inside cells it can be repackaged or it can be inactivated by monoamine oxidase in the mitochondria. Some of the dopamine would leak out into the bloodstream وبيصير له transfer or transport into the liver, where it gets inactivated by also these two enzymes أنا بنحكي عنهم, or it can be taken up by neighboring cells including the postsynaptic neuron, where it can also be inactivated. In terms of the re-entry of dopamine inside the presynaptic nerve, it takes place via a transporter, sodium ion dependent transporter, and this is the target of cocaine and amphetamine, leaving dopamine in the synaptic cleft for a longer time so that it can induce excitation, energy, and the person would be like fully aware and excited, in other words. هذا بالنسبة للدوبامين. النورابينفرين بصير في عنا packaging of dopamine inside vesicles, وهاي الفيسيكلز بيصير فيها conversion of dopamine لنورابينفرين. طيب وبعدين again calcium ion influx, fusion of vesicles with the plasma membrane, release of norepinephrine into the synaptic cleft, ونفس البروسيس of inactivation أو termination of signaling اللي هو reuptake, uptake by neighboring cells, diffusion, enzymatic inactivation, and so on. أما بالنسبة للإبنفرين إنه في عنا النورابinephrine now is now inside vesicles. It leaks out of these vesicles and it gets converted للإبنفرين. And then epinephrine is packaged again inside vesicles. ونفس الحكي fusion, release, reuptake, uptake by neighboring cells, diffusion into and transport to the liver, enzyme inactivation, and so on. هلا بالنسبة للpackaging of these neurotransmitters أو the catecholamines inside vesicles, خصوصا بنحكي على الدوبامين والابنفرين, these are transported inside vesicles via an ATP-dependent process that requires also the efflux of protons from the vesicles themselves. فاللي بيصير عندنا actually إنه we have a transporter, a vesicular ATPase transporter that consumes ATP for the entry of protons inside vesicles. So the protons, the vesicles are acidic inside, low pH, and there are a lot of positive charges inside cells versus negative charges outside in the cytosol. فبصير في عنا exchange, transport as well between the protons and the monoamine neurotransmitters. And this transporter right here, the VMAT, the vesicular monoamine transporter, can be targeted by certain inhibitors or drugs, leaving the monoamine neurotransmitters in the cytosol for a longer time, where they can be inactivated enzymatically, and that causes depletion of these neurotransmitters. نتكلم على inactivation of these neurotransmitters. So it happens enzymatically. We're talking about the enzymatic inactivation. It happens via two enzymes. The first one is known as monoamine oxidase. The other is known as the catechol or methyl transferase. It doesn't matter which one starts first. يعني ممكن المونو امين اوكسيديز وبعدين الميثيل ترانسفيريز، او ممكن ميثيل ترانسفيريز بعدين مونو امين اوكسيديز. Since we're talking about a methyl transfer reaction, فعشان هيك احنا بحاجة لفيتامين B12 and the folate عشان for the synthesis of the S-adenosyl methionine inside cells. طيب الاشي الثاني as well انه these enzymes exist in the presynaptic neuron as well as adjacent cells مثل الglial cells والpostsynaptic neurons as well. وعندنا drugs حكينا هذه previous slide انه drugs that result in inhibition of the transfer or the transport of these neurotransmitters inside vesicles causes depletion of these neurotransmitters لأنه هدول enzymes would act on these neurotransmitters, degrading them, inactivating them. Something else إنه بالParkinson's disease لما يكون عندنا high level of dopamine that results in the presence of homovenilic acid اللي هو ال end product بالurine. فهدول the drugs would result in the inactivation of the dopamine with higher level of homovenilic acid in the urine. But people with Parkinson's disease, they have reduced level of the homovenilic acid in urine. وبالنسبة للregulation حكينا انه اول reaction اللي هو tyrosine that is catalyzed by tyrosine hydroxylase هو ال rate limiting reaction لانه هذا الانزيم is regulated and it is regulated short term and long term. Okay, فعندنا short term regulation and long term regulation. Long term regulation على طول we're talking about gene expression, we're talking about transcription with translation and it takes time, takes hours. فلما يكون عندنا prolonged sympathetic neuronal activity لما يكون عندنا a lot of firing بالneurons حيصير في increased transcription of tyrosine hydroxylase as well as dopamine hydroxylase اللي بيحول dopamine إلى norepinephrine. ال short term regulation بصير at the level of enzyme activity فلما يكون عندنا high level of cytosolic catecholamines they will compete with the tetrahydrobiopterin binding للإنزيم فبصير في عندنا inhibition of the enzyme. In addition ممكن يصير في activation by depolarization فلما يكون عندنا a lot of depolarization حيصير في increased activity لبروتين كاينيز C, بروتين كاينيز A, calcium calmodulin dependent kinases اللي بيعملوا dephosphorylation اللي بيعملوا phosphorylation للتيروسين hydroxylase and as a result activation لأنه بصير في عنا increased binding increased affinity between tetrahydrobiopterin and the enzyme. فاحنا حكينا من شوي عن التيروسين derived neurotransmitters. نتكلم هلا على التريبتوفان derived neurotransmitters وعندنا اثنين اللي هم تريبتوفان اللي هم سيروتونين سيروتونين and melatonin. هلا what happens انه التريبتوفان would be hydroxylated للفاينل product اللي هو سيروتونين. هذا لأنه إحنا عمالنا بنتكلم على hydroxylation of a ring فإحنا بحاجة إلى tetrahydrobiopterin. بصير في packaging of serotonin into vesicles برضو by the vesicular monoamine transporter بنفس الميكانيزم اللي حكينا عنها في السابق. Again, influx of calcium ions, fusion of the vesicles with the plasma membrane, release of serotonin ونفس الميكانيزم of inactivation اللي هو reuptake, uptake by neighboring cells, enzymatic inactivation أو diffusion as well. طيب فبكون عندنا الفاينل product اللي اسمه hydroxyindole acetic acid which would be present in the urine. So antidepressants زي اللي هم selective serotonin reuptake inhibitors target this transporter right here, prolonging the presence of serotonin بالsynaptic cleft prolonging its activity or its action. This is the mechanism of action of these drugs. بالنسبة للميلاتونين it is synthesized من السيروتونين. Serotonin would be converted to melatonin via methyltransferase enzyme. Melatonin, by the way, is important for sleep pattern, seasonal circadian rhythms, يعني day and night, for example, seasons, fall, summer, winter, and spring. هلا بالنسبة للأمينو acid based neurotransmitters, it just happens إنه ممكن some of these amino acids would act as neurotransmitters themselves, أو إنه they would be modified a little bit, resulting in the generation, production of a neurotransmitter. An example is histamine. Histamine cannot penetrate the blood-brain barrier, and that's why it has to be synthesized in the brain. Histamine is inactivated by two enzymes اللي هم الhistamine methyltransferase and diamine oxidase أو histaminase. Histamine is packaged into vesicles via VMAT as well, the same transporter as the serotonin, melatonin, and the catecholamines. Synthesis takes place by decarboxylation of histidine, resulting in the production of histamine. This reaction requires pyridoxal phosphate, vitamin B6. One thing important about histamine is that it's not recycled. It's not taken up by the presynaptic neuron, and this is a difference between histamine and the other neurotransmitters. In addition, the way it gets inactivated is by monoamine oxidase in astrocytes. In terms of glutamate and aspartate, both of them are non-essential amino acids. We can synthesize them ourselves. They do not cross the blood-brain barrier, and that's why brain and neighboring cells can synthesize these two amino acids. And in addition to neighboring cells like glial cells, they can also synthesize these amino acids. Both are excitatory neurotransmitters. In terms of synthesis of glutamate, عندنا three mechanisms or three pathways. The first pathway اللي هي عن طريق الجلايكوليسيس. Glycolysis, Krebs cycle, بيصير عندنا alpha ketoglutarate اللي بيصير له transamination reaction بنحط له amino group فبتحول إلى glutamate. So that's the first mechanism. The second mechanism is via deamination of glutamine. فباسically احنا بنشيل الامينو جروب من الجلوتامين بتحول إلى glutamate. And the third is transamination of aspartate involving برضو alpha ketoglutarate, resulting in the production of glutamate. In terms of removal, inactivation of the glutamate, it has to be taken up by cells, either the presynaptic cells or by the postsynaptic cells or the by glial cells as well. طيب فلاحظوا انه بالglial cells if glutamate is taken up by these cells, it gets converted to glutamine. Glutamine is transported out to the presynaptic neuron where it can be converted to glutamate. This here just to show you the process. فعندنا الالفا كيتوغلوتريت ممكن ياخذ امينو جروب من الاسبارتيت او actually ممكن الجلوتاميت ديهيدروجينيز اللي هو الجلوتاميت ديهيدروجينيز فبتحول الى glutamate. Glutamate can then be converted to glutamine. And this is the cycle that you learned before in the metabolism course. So, in terms of aspartate, في controversy إنه هل الأسبارتيت neurotransmitter ولا لا. لكن what all we need to know about it is that it gets synthesized from oxaloacetate by transamination reaction. So involving alpha ketoglutarate and glutamate as well, and aspartate transaminase. In terms of glycine, it is an inhibitory neurotransmitter. In contrast to glutamate and aspartate, it is synthesized from serine by serine hydroxymethyltransferase وعن طريق ال 3 phosphoglycerate اللي هو intermediate بالglycolytic pathway. فاحنا عندنا جلوكوز ل 3 phosphoglycerate لسيرين ومن سيرين إلى glycine, and it gets inactivated or termination of signal by high affinity transporter, either by the presynaptic neuron or neighboring cells as well. GABA is the abbreviation for gamma aminobutyric acid. It is present in high concentrations in certain brain regions. Concentrations can be 1,000 times higher than concentrations of classical monoamine neurotransmitters, and there is something known as the GABA shunt, a metabolic pathway to conserve the supply of GABA. So the way it is synthesized, either by glutamine لglutamate وبعدين glutamate to GABA by glutamate decarboxylase. So this pathway shows the decarboxylation of GABA, decarboxylation of glutamate to synthesize GABA. And then GABA itself would be stored inside vesicles. Oh, by the way, the decarboxylase enzyme requires vitamin B6 as well. GABA is stored in vesicles and it gets released once you have influx of calcium ions. And GABA is then either taken up by the presynaptic cells and repackaged, recycled, or it goes into glial cells where it gets converted to glutamate. Okay, it gets converted to glutamate, and then again glutamate to glutamine, and then glutamine is transported out of glial cells to the presynaptic neuron, and it gets converted to glutamate and to GABA again. So this is the GABA shunt. In terms of acetylcholine, acetylcholine is synthesized from choline, which integrates or condenses with acetyl coenzyme A to produce by choline acetyltransferase to produce the acetylcholine. So where do we get choline from? We get it either from diet or we get it from the plasma membrane. Okay, so phosphatidylcholine, for example. It gets transported and stored inside vesicles, and then the same story, vesicles fuse with the plasma membrane, and once acetylcholine is released, it gets inactivated by acetylcholinesterase, which is the target of SARS, okay, the neurotoxin compound used in wars. So this is how acetylcholine is inactivated. It gets converted to choline and acetate, and choline can be taken up by cells again. نتكلم على nitric oxide finally. Nitric oxide is a gas, and the definition of neurotransmitters اللي أنا حكيته في أول المحاضرة does not apply على nitric oxide. فnitric oxide is an exception. فنبلش كيف بيصير synthesis للنيتريك اوكسيد. هلا الجلوتاميت بيصير له packaging inside vesicles ولما تيجي signal depolarization وتيجي signal بيصير في release للجلوتاميت into the synaptic cleft. Some of it would be and it would bind على receptor اللي هو N-methyl D-aspartate receptors الموجودة على postsynaptic neurons. هلا what happens انه once it binds to these receptors, calcium ions would go into the postsynaptic neuron, activating nitric oxide synthase inside cells. Nitric oxide synthase, once activated, converts arginine to nitric oxide. هلا nitric oxide then stimulates guanylate cyclase اللي راح ايش راح يعمل لي؟ حيعمل لي cyclic GMP from GTP. cyclic GMP can then bind to many proteins and enzymes, resulting in a physiological response. هلا nitric oxide نفسه can diffuse out of cells, and it can enter neighboring cells like glial cells and other neurons, and it can diffuse into the presynaptic neuron. بالpresynaptic neuron مرة تانية nitric oxide can activate guanylate cyclase to produce cyclic GMP. Again, it binds to different proteins, eliciting a physiological response. طيب فعشان هيك هذه احنا بنسميها retrograde pathway او بنسميه retrograde messenger. ليه؟ لأنه ممكن يرجع انه يرجع مرة ثانية للpresynaptic neuron, inducing a signal. Its half-life is 2 to 4 seconds, so short duration, and it can be inhibited by binding to proteins like hemoglobin and other heme-containing molecules. So it has a very short half-life, لكن أفعاله ظاهرة ومبينة كتير. And it can be inactivated by diffusion as well. طيب السؤال هو, is nitric oxide a neurotransmitter? And the answer is yes, ولكن pay attention, it is not stored in vesicles, it is not released by calcium-dependent exocytosis, its inactivation is passive, يعني ما في active process مثل ال reuptake, لا هو diffusion via the plasma membrane. It decays spontaneously by itself. It can be inactivated and it does not interact with the receptors على cell surface. Rather, it goes inside cells. It binds the enzyme اللي هو guanylate cyclase to produce cyclic GMP. Its sphere of action depends on the extent to which it diffuses. لو يمكن يوصل, لأنه احنا بنتكلم على very short half-life, 2 to 4 seconds. يا دوب يطلع من السيلز ويعمل interaction مع neighboring مع enzymes ب neighboring cells. It acts as a retrograde messenger. It regulates the function of axon terminals بال presynaptic neuron as well. نفس النيورون اللي اشتغل عليه. So, and differences. So, nitric oxide synthase, by the way, عندنا 3 isoforms. I do not want you to know كل واحد منهم ايش هو, لكن I need you to know انه isoform 1 هو اللي موجود في النيورونز وهو اللي ممكن يكون target ل different drugs. What's important is that all 3 isoforms require dihydrobiopterin as a cofactor, as well as NADPH as a coenzyme. والله معكم.